will allow development of immune strategies to inhibit tumor microenvironment, immunosuppression, and improve patient outcomes. The recent study demonstrates the role of immunosuppressive neutrophils in the pathogenesis of multiple myeloma. This study is an example of bedside to bench to bedside translational research leading to a phase 1 study examining CXCR2 inhibitors of neutrophils along with standard salvage therapy for relapsed refractory myeloma. They continue to develop translational research projects that will develop into novel therapies and advanced treatment of cancer with emphasis on multiple myeloma. Thank you very much, Philip, to joining us. And yeah, please proceed with your presentation. Thanks.
Thank you, Vitaly. I really appreciate the invitation to speak. Hopefully I'll be someday back again in Cork to visit. And let me see if I can get this to, there we go. These are my disclosures of institutional support, which is finally, I think, going to run out from originally from Celgene and then later on BMS, Beijing advisory panel, as well as GlaxoSmithKline. And I will be discussing non-EMA and non-FDA approved indications during my presentation. So I don't know, this is for older people, but for those of you who have seen the film, The Princess Bride, it was featured as part of an editorial by Sagar Lonial, who's chief of hematology at Emory, along with Anita D'Souza. And they were talking about the tourmaline study, which was ixazomib Lendex versus Lendex. And what they quoted was from the film itself, where Vizzini, the character played by Mandy Patinkin, has an exchange with Inigo Montoya, who plays a bad guy. Vizzini, I got it wrong, is Wally Shawn. Inigo Montoya is Mandy Patinkin, who started off as a bad guy, but turned into a good guy. And this is a film from 1987, directed by Rob Reiner. May rest in peace. And Vizzini keeps saying 5 times total, inconceivable when faced with the opposite of what he thinks should happen. And hearing this for a third time and seeing the reverse, Inigo Montoya says, I do not think that word means what you think it means. And how does this relate to myeloma? 20 years ago, it was inconceivable that the median overall survival for a myeloma patient would be greater than 10 years. We're now, for those who receive a transplant, approaching 20 years. And to consider alkylator-free induction regimens for fit or frail patients, and that post-relapse outcomes could be almost as long, in some cases longer, as first responses in phase 3 trials would be confounded by post-salvage therapies, including at the time, daratumumab, which wasn't necessarily frontline, but now is, carfilzomib, and B-cell maturation antigen-directed treatments. Yet, that is where we are in the field using modern myeloma therapy. So, I put this up. This is adapted from Cancer's article from a few years ago, and then Jesus Barreda at ASCO. And the myeloma cells in the middle, and surrounded by all the things that can kill it. And we can start over here with NK cells, which are modulated by both DARA and ESA, as well as having a direct anti-myeloma effect, elotuzumab targeting SLAMF7, and then the CELMoDs and which are new and will be hopefully approved later on this year, as well as the IMiDs, which have been a mainstay of therapy. At the bottom is the anti-BCMA ADC belantamab, and which was then reapproved, fortunately. The bispecific T-cell engagers, which are in common use now, targeting both BCMA and GPRC5D. And then at the very bottom, here's the CAR T-cell, which will be the focus of my talk. And then on the right-hand side, you can see all the potential targets that have been developed or in development for treating multiple myeloma. As it turns out, BCMA is not going to be the final answer. So this is from the NCCN guidelines, which I just took out or took screenshots of. And this is the initial approval after 3 prior lines for treatment in myeloma. You had listed cilta-cel and ide-cel. And then the bispecifics for 4 prior lines. Now, it's moved up. So going from 1 to 3 prior lines, we now see that both cilta-cel, category 1 for first relapse, and then ide-cel after 2 prior lines. Things have changed. So this is I'm not going to go through this in great detail. This is just a summary of how CAR T-cell developed over time. Back in 2007 with the first IND investigational new drug against CD19, and then development of co-stimulatory molecules to make it work better. We're still at second generation with either CD28 or 41BB. Third generations and beyond have not been fully developed. And then the different companies I'll list up here. The top is Novartis with their ALL and lymphoma drugs. To the right is Kite Gilead with lymphoma as well as ALL. At the bottom in the middle, you have BMS with a variety of indications from CLL to lymphoma. And at the very bottom is ide-cel in red. I won't talk a whole lot about ide-cel because the field has moved more towards using cilta-cel, but I'll show that a little bit later. And then over on the right-hand side is Janssen's cilta-cel in partnership with Legend. Initially, both of these were for 4 prior lines, but now cilta-cel's after 1 prior line and ide-cel after 2. And then at the bottom here is obe-cel, which is Autolus's ALL drug. So I took this screenshot from something called, oops, my daughter's calling me. I'll have to call her later. This is from Sleuth Insights, which is, you can go on the web and do actually downloads. And this is a listing back in 2022 of all the products in development around the circle. And in the middle, it's hard to see, are all the approved indications back in the US. Red is allo and green is auto, and there's a different way of, some are TIL, some are TCR based, some are CAR T, some are NK cells and macrophages. So as you can see, there's quite a bit of interest in this type of treatment. Then here are the in vivo CARs, which as you can see have not moved into the center, which would be FDA approval. They're all on the periphery, but they're currently in development, and there's quite a bit of interest in that, and I'll show a couple of instances of that. And here you have red is oncology, green is inflammatory, such as lupus or rheumatoid arthritis, purple's both, and then yellow is other. And the target we're going to focus on a lot today is BCMA, member of the TNF receptor family, and a ligand, the receptor, for the APRIL and BAFF, B-cell activating factor, and which are and both of those are proliferation signals which cause plasma cells to grow. And you can see on the top in red are the expression of BCMA, which becomes quite heavy when the tumor becomes more prevalent, as well as when the tumor is malignant. And plasma cells, while they express it, do not express it as large a degree. BCMA is also excreted. I won't talk too much about that. There was a study looking at a gamma secretase inhibitor, which is what cleaves BCMA off, but that still is not ready for prime time. So this is sort of the scheme of what happens with CAR T-cell therapy. I'm sure most of you are familiar, where there's a screening process. There is to see about patient eligibility. There's leukapheresis, where the cells are collected and then manufactured. And there's a variety of different ways that occurs, with autologous especially. And then there's a conditioning regimen, usually fludarabine, cyclophosphamide, occasionally bendamustine. And this occurs after the cells have been manufactured, sent back to the site. There's a T-cell infusion, and there's either close observation, outpatient with daily visits versus standard hospitalization, and that varies depending on toxicity, and then there's follow-up and tumor assessment. So this is a slide courtesy of David Porter. It does have animation, and this is the cartoon showing how CAR T cells work. In the lower left hand, you can see the chimera, which is goat, serpent, and lion. All cultures have a chimera, and thus the term chimeric antigen receptor. And then it involves gene transfer stably into of the CARs into T cells, and the CAR uses the cytotoxic potential of the T cells to kill They need to persist, as well as be effector cells. And fortunately, these are non-cross resistant to chemotherapy. The first trial back in 2011, its publication in NEJM by David. And then you can see these are the list of other publications for both ide-cel and cilta-cel, which led to the development of the BCMA CAR. And this is how it works. It's usually a lentivirus that's T-cell trophic, puts the CAR into the T-cell. It's expressed on the cell surface and seeks out and destroys anything that expresses BCMA. Excuse me. I'll go through this fairly quickly. These are all the CARs that are currently FDA approved. The first one was Tisa-cel, which is a murine construct anti-CD19. So anyone who gets this is a mouse-human hybrid. Here is the axi-cel and bruxa-cel from Kite Gilead. This is BMS's, which initially was murine, but then was humanized. And this also targets CD19, Lysocell. And then there's obe-cel, which was just last year approved for ALL. And then we move now to the two myeloma drugs or cell products. The first one is ide-cel, which is a murine construct anti-BCMA. And for those of you who don't know, llamas only make heavy chains. So this is a llama human hybrid patients who receive this. So the only other species that makes only heavy chains are sharks. I was wondering why they didn't do that for marketing purposes. But anyways, this was developed by Legend Bio, and then in partnership with Janssen led to the development and approval of this drug. Now, what's currently on the horizon, although it keeps getting delayed, with change in leadership in the FDA, they changed how they want to approve things. So it was expected that anito-cel would have been approved by second quarter, but they were given a PDUFA date, which is the Prescription Drug User Fee Act, which essentially stands for the FDA has to make a decision by December 23rd, 2026. That's at the bottom there for a biologics license application. What's interesting about anito-cel is it's a computer design, so it's this little nubbin here, and it is different. Here's cilta-cel with the two heavy chains, and then the light and heavy chain for ide-cel. And its small construct allows for more expression of this CAR on the cell surface, and it's different in that it's very stable. It will not fold apart. It stays together, and because of its small size, it has a less likelihood of tonic signaling. The thought is that binding and sticking or increased tonic signaling leads to increased toxicity, and this doesn't seem to have the toxicity of cilta-cel. So Poseida, this is also showing that Arcellx was acquired by Gilead just recently. So they're really betting on this to be the next great thing. And in the meantime, there's an AlloCAR Poseida that was acquired by Roche, and then Colonia, which I'll talk about a little later, has a collaboration strategic partnership with J&J. Also Linkcell, which is a different form of technology to allow for safe tumor killing and targeting multiple antigens. And this is also a screenshot from Sleuth Insights showing all the different companies that have been either taken over or developed partnerships or over the time for the last not even 15 years. It's quite a big field. And this also shows just how CAR T-cell therapies have increased in number. This is from the transplant registry based in Milwaukee and Minneapolis, and this looks at the number of autos here in dark blue. The purple are the allogeneic transplants, and here in lighter blue are the number of CAR T cells over time. And this is breaking them down by cell type. And you can see in gray are the number of large cell lymphomas. But here are the myelomas in blue and rapidly rising because of the need, because of the current so-called not curability of myeloma. And this is also a screenshot showing the current up to 2023, and then this is behind, they haven't updated for 2024, but the estimated number of cilta-cel here in gray, ida-cel are the ones in right below it in this purplish color, and then below here in blue are the lyso-cel. This is Yescarta, axi-cel, and then bottom is Kymriah or tisa-cel. So this is a rapidly progressing field, and I don't expect, though, the numbers of cilta-cells to continue like this based on estimates. And this is data both on the in vivo CARs as well as anito-cel. So CAR T-cell toxicities. These are the major issues that occur, and we have to be thinking about all the time. The cytokine release syndrome is pretty well managed now, as long as we understand what's going on. I like to say that initially treating patients with CAR T cells was like giving a Maserati to a 14-year-old. You didn't quite know what you were doing, and then there was a steep learning curve, which we've now been able to manage for CRS. ICANS is a little different, and I can talk a bit about that in a sec. And there's also hematotoxicity, which I'll also go over. And there's here on the right are a variety of inflammatory markers that get increased. We now know that tocilizumab, an anti-TNF receptor antibody, is a mainstay of CRS treatment. For neurotox, less so tocilizumab, more steroids, high-dose dexamethasone, as well as anakinra. And for severe cases, we actually use ruxolitinib because of its ability, as well as anakinra, to cross the blood-brain barrier. So this is a very simplistic way I put it, that CAR T-cell is similar to an auto transplant in that the major toxicity occurs and resolves by day 30, with some exceptions. Lymphodepletion and cell infusion occurs, although the lymphodepletion is not as intense as the therapy for an auto transplant. And there's a whole issue of inpatient versus outpatient and hospitalized for toxicity, which isn't always predictable. And a lot of our patients get moved to the inpatient service if they're having a more severe toxicity, especially ICANS or severe CRS. And prior comorbidities often affect outcome, and I'll discuss that a little bit later. And that CAR T-cell therapy is very different from an auto in that the products are ridiculously expensive. They're over $500,000 just the product alone in the US. There's education for recognition and treatment of toxicities, which are not always predictable. And the big things that we worry about are tumor lysis syndrome, and high tumor burden obviously predisposes to TLS, but it also predisposes to the cytokine release syndrome, ICANS, and motor neuron disorders, which I'll mention briefly. CRS, as I mentioned, can be fever, hypotension, but it can also be quite severe, recapitulating or mimicking HLH or macrophage activation syndrome. ICANS is usually altered mental status progressing to encephalopathy if it's not treated adequately in coma. So we don't want, obviously, that to happen. It can occur with and without CRS. And there's a motor neuron disorder that occurs usually after cilta-cel, occasionally after axi-cel. That's also dependent on disease burden, as well as having prior neurologic disease, not necessarily cancer in the brain, but other neurologic disorders such as a prior stroke. And now there's the immune effector cell-associated hematotoxicity, which is not quite clear how it works, but it does cause prolonged cytopenias after CAR T-cell therapy. And there are risks for infections due to the prolonged hypogammaglobulinemia, as the normal B cells are innocent bystanders and often are eradicated, or not eradicated, but knocked down by the CAR T-cell. And I do like to say, when you've seen one CAR T-cell patient, you've seen one CAR T-cell patient. They don't always follow a straight playbook in terms of toxicity. This is from a EBMT IMWG consensus article talking about the need for infection prophylaxis, comparing EBMT, IMWG. And the big issue is a lot of patients, as I mentioned, are hypogammaglobulinemic, and that immunoglobulin replacement is often a mainstay, as well as early antibacterial prophylaxis, which is then removed. Same with antifungal. We do pneumocystis prophylaxis, especially if the CD4 counts are low, and that can occur for quite a while, for several months afterwards, before the CD4 counts come up. Also, it's not clear if the CAR T cells are pushing out normal T cell development. The thing that's very interesting is last year, the FDA eliminated the REMS program, Risk Evaluation and Mitigation Strategy, for CAR T. And they removed a lot of the requirements, such as having on-site tocilizumab and certified hospitals, which was usually done by the companies. And the post-infusion monitoring was reduced from 10 to 7 days. After infusion, the required patient stay near the healthcare facility was reduced to 2 weeks, as well as the driving restriction. And this is quite novel, and it made sense because a lot of the CAR T cell therapies did not have prolonged toxicity. That was a big concern initially. So this is outcomes. So this is a comparison. This is in JCO from 2025. Our group participated in this. And the ide-cel is in red, cilta-cel is in blue. On the right-hand side, you can see the PFS based on intent to treat, superior for cilta-cel as well as the OS. And if you look at the PFS infused, it's also similar. The interesting thing is, if you look at the initial curve for OS, you'll see that it dips for cilta-cel, and then ide-cel gets worse based on disease progression. And this is the toxicity associated with cilta-cel, which is greater than it is for ide-cel. So this was presented at the ASCO meeting last year, and it's a picture of the skull, and it's made the New York Times, which made it sound like everybody was going to get cured, which was not quite true. And this is a recapitulation of what I showed earlier, where here you have the eligibility. These were patients who had 3 or more prior lines, apheresis, bridging therapy, lymphodepletion, and then cilta-cel with a fixed dose as opposed to cells per kilo, which some of the other products use, and then a long-term follow-up. We still need to do a 15-year post-infusion follow-up, and that's for commercial products as well. It used to be twice a year, now it's down to once a year. This is the results showing that indeed the PFS was 35 months, OS was not reached at 33 months, and this was compared to a heavily pretreated group called the Locomotion from the European group, Dr. Mateos' study, showing that the median PFS was less than 6 months and the median OS was approximately 1 year. And this is showing that the 5-year progression-free survival was approximately 33%, showing that indeed some of these patients still had not disease progression after 5 years and potentially were reaching a plateau. And this is showing their future studies. That was CARTITUDE-1, which I showed you. CARTITUDE-4 shown here on the right-hand side for both standard and high risk compared to standard of care therapy, showing the benefit for cilta-cel. And that's leading them to do future studies looking at it in frontline. This is 1 to 3. There is a transplant ineligible study looking at RVD induction plus RD versus RVD and cilta-cel. And then CARTITUDE-6 will be looking at the quadruplet, followed by auto transplant, and then versus cilta-cel. Now, will CAR T cells control the landscape? Not clear. This is the late-breaking abstract by Dr. Mateos at Majestic. I downloaded these from what is called CCO now, is this Sarah Clinical. And this is comparing Dara, teclistamab, and anti-BCMA, and daratumumab versus DPT, DVD, dealer's choice for investigator's choice for salvage therapy in 1 to 3 prior lines of therapy. And this is the 36-month PFS is 83% versus about 30% for the DPT-DVD. And this is comparable. It does mean therapy every month, but it is comparable to cilta-cel. Of course, you're not supposed to compare studies without a head-to-head. But it is intriguing as to what will be the possibilities, and can we start thinking about using bispecifics in different situations, say after a CAR T cell therapy. So I'm just going to show some selected studies to improve CAR T cell therapy and expand indications. This is the EMN 28, the CARTITUDE-6, and this study is accrued. 750 patients. The MRD is the readout, or so the primary study objective was secondary of PFS and OS, and the readouts is expected between 2027 and 2028. That's one problem with phase 3 studies. It's a good problem for the patients. It's harder for the investigators because you have to wait for the readout because patients do so well. And in this case, this is a quadruplet induction followed by transplant, 4 versus 6 cycles, of which you then follow with cilta-cel. And in Europe, there's more interest in a consolidation. So 2 cycles of RBD. Everyone gets R maintenance and then follow up until PD and then long-term follow-up. The thing is, there's no DARA as part of the maintenance, but the major focus on transplant versus CAR T-cell therapy. And this is a very busy slide. I apologize. And this is looking at anito-cel, which has a similar PFS in their phase 1/2 studies to cilta-cel. It's about 33 versus 36 months and much less toxicity, at least initially in terms of a particular ICANS. And this is this is provided by Dr. Mateos. This is the GEM Anito first. It's a small sort of proof of principle looking at 12 patients per cohort. Cohort 8 is the quadruplet times 3 cell collection, and then a quadruplet times 3, followed by anito-cel and then len maintenance. Cohort B is a transplant ineligible group, which is ESA VRD, leukapheresis, 2 more cycles, and then anito-cel with len maintenance. And cohort C is very similar, except there's no maintenance. So this has just started enrolling and will then guide them in terms of larger clinical studies. Another way is the CD19 BCMA combined CAR Ts. And the bottom left shows the theoretical reason for doing this. This is from a paper from our colleagues in Germany, showing that CD19 expression does occur. It's more in the MSCs, and so this may be a way to target the stem cells as well as on newly diagnosed or relapse refractory, target them with BCMA. The Autolus group has done a combined BCMA CD19 CAR T from UK, and that's currently in trial for myeloma. They're moving up the indication from heavily treated to early, and then they also have an amyloidosis study with this drug. There is a study that came out of China. This is looking at the bispecifics. There are different types of them, and this is showing the PFS on the right. And then AstraZeneca has partnered with Gracell, I think they actually bought them, to look at a combined CD19 BCMA, which is currently enrolling both the phase 2 and then they're starting a phase 3. The interesting thing is they have a fast CAR, so to speak, where it's 48 hours for the manufacturing of the CAR, which is very different than the current manufacturing, which can be a few weeks. And then can you make the response to a transplant improved by using CAR T-cell followed by maintenance? This study was presented at the transplant meeting in 2025. And this is for patients who are less than a CR after an auto transplant. This at about 6 months, they would undergo leukapheresis with then manufacturing of ide-cel, followed by LD chemo, and then a CAR T-cell infusion, and everybody gets len maintenance if tolerated. And this is looking at the rate of best response at 6 months, showing that there are a large number of patients who attain CR. And then this is the swimmer's plot at the bottom, showing that so far there's only been one reported progression, and everyone else is doing fairly well. So this is another possibility. It's not clear if you would be doing this with ide-cel because the field has moved towards using more cilta-cel, but we'll see. So this is Arlo-cel. This is a GPRC5D directed CAR T-cell. BMS is running this. And the same thing with the lymphodepletion. The toxicity is fairly reasonable with CRS and not too much neurotoxicity. And then the extended follow-up showing that the overall response rates are good. And they now have 2 studies, the phase 2 quintessential, which is in triple and quad refractory patient, or sorry, exposed patients. And then the quintessential 2 is the comparing Arlo-cel versus standard of care in 1 to 3 prior lines. So stay tuned for that one. And this is a T-cell receptor therapy. I sort of stuck a bunch of things together. And this one's actually approved afami-cel for synovial sarcoma. And in this case, what here is done, and it's hard to see in the small one on the upper left, is that the patient's own T-cell receptor is removed and a TCR is put in that recognizes MAGE. And in this case, it's an antigen that's associated with sarcoma. So you can see here on the bottom left, you now are recognizing not a tumor antigen, but a peptide that's associated with class 1. So it's used in 3 different class 1 situations, class 1 HLA-A, and they based it on ethnicity. So some are seen more in white versus black versus Asian populations. And this is a cartoon which I got from a laboratory in the Netherlands showing here the protein, it's cleaved by the proteasome, it's processed and then placed on the cell surface in the context of class 1, and the CTL comes over, recognizes it, and kills it. And so for CAR therapy, the specialized receptor, or CAR, is synthetic. TCR therapy uses a modified form of a naturally occurring TCR. And they target, as mentioned, the TCR therapies, abnormal proteins found inside the cancer cell, which gets moved to the cell surface as opposed to a CAR that recognizes something such as BCMA. And this is the Kaplan-Meier curve of the responders, which always do better than non-responders. The green are the overall population, the red are the non-responders. So stay tuned, as there may be some peptide-associated TCRs for multiple myeloma. Auto versus allo, I say have about 10 minutes. So this is a step showing what needs to be done to make an auto, which I went over earlier. The cells are collected, manufactured, and brought back to the patient for use. The allos are made by taking a normal person's T cells and then engineering them so that they don't cause problems such as graft versus host disease or early rejection. And then they're put in the freezer so they can be pulled out and used at any time. And I grabbed one of the results presented by Poseida. Poseida was bought by Roche. And that, so they're interested in that one. And this involves an engineered allogeneic CAR. Up here on the right shows the persistence in several patients over time. This on the lower left are some of the cell lines that are killed by this allogeneic CAR. And this is a waterfall plot showing the drop in M protein, or the change in myeloma parameters, I should say, over time in this patient population. So we're participating in this. They have some interesting things where they're trying to, they put in actually a dihydrofolate reductase and then use methotrexate to try and enhance the persistence of the allogeneic CAR against BCMA. And then in vivo CAR, which is kind of really interesting, which involves taking something in the laboratory, essentially is taking a virus-like particle, which used to be a virus, gutting it, and then putting in the molecules of interest to make an in vivo CAR. Here on the right is the comparison of all the things that could be done to enhance efficiency as well as prevent rejection and graft versus host disease with the allo CARs. And this is one that was published just recently in Nature Medicine. The first 5 patients, and this is a company in China that's developed this, and you can see that for the 4 patients who are alive, that 3 had attained a stringent CR, 1 is still a VGPR, and but MRD negative, interestingly enough. And 1 patient died early of sepsis, and that you can see here in yellow where everything stops. So there was a lot of concern about the toxicity, because even in 5 patients, this 1 patient who died and the others who had CRS and also some ICANS. So this is the one that was presented at ASH as a late-breaking abstract. These slides are courtesy of Dr. Joy Ho. And this is also a more detailed look at the virus-like particle. I don't have all the slides from this. It's quite interesting, however. And so they put a CAR in, they put a molecule on, also computer designed, that will target and bind and allow introduction of the modified particle into the T cell. And they also have detargeted something called the fusogen, which is an LDL receptor. So it prevents it from being non-specifically taken up by reticular endothelial cells. So this allows for precise targeting to T cells. And also based on the CAR that was developed, good binding to BCMA. So I squeezed all these slides together. This is the lead or introductory slide. And then you can see here on the upper right, the T cell expansion was commensurate in the 5 patients they studied with approved CAR T cell therapy. It's hard to see it, but they had data from the first 3 patients. They're very similar to what you would see with CARTITUDE-1 and CARTITUDE-4. Interesting, patient 3 had a very robust, it's this red thing. And so they got very nervous. Sorry, it's the green one. So they got very nervous and they actually went to dose minus level 1 because they were concerned that they would have too much robust T cell expansion. As it turns out, the 4th patient also had a lot of expansion. So even at the -1 level. And then this is on the lower right, shows the toxicity. There's essentially no ICANS in these 5 patients, and all the CRSs were grade 1 to 2, and they had the usual cytopenias, lymphopenias, which occur afterwards. And actually, somebody had a fainting episode, but everybody's still alive. And these are on the lower left, what's going on in terms of the first patients, the one furthest out, just to 6 months, showing that the patients in CR. All patients, the first 3, I should say, attained MRD negativity at 10 to the -6 at day 30, which is quite interesting, before the protein had even gone away. And the 4th patient was too early. It was MRD negative by flow. And this is just showing the serum-free light chains dropping, the M spike levels, and the soluble BMA also dropping off. So this is a very exciting new possible product. As I mentioned earlier, Johnson has made an agreement with Colonia to bring this forward into further clinical trials. So why do the majority of CAR T cell and lymphoma patients, lymphoma and myeloma patients relapse? There's a variety of reasons: limited anti-tumor activity by the T cells initially and over time, targeted antigen loss. I didn't go over that, but BCMA can be deleted genetically. Target antigen shedding and interference with CAR T cells, T cell inability to reach tumor, limited trafficking, and then immunosuppressive tumor microenvironment. Myeloid-derived suppressor cells, tumor-associated macrophages, inflammatory and suppressive cytokines, hypoxia and altered glycolysis, and then diminished T cell metabolism in fitness, sometimes seen with exhaustion. Exhaustion is seen with chronic viral infections where the T cells have chronic antigen exposure and essentially stop working. So what do we need? We need better CAR T cells, other effector cells such as NKs and macrophages, better targets, earlier CAR T cell indications, maintenance therapy, which is very interesting. In fact, we have one that's proposed to the Alliance Cooperative Group looking at mezigdomide, actually to enhance lymphocyte manufacturing. We're also interested in using it as maintenance, although it is pretty hematotoxic, so we'll have to see. We do know that patients who receive bites before CAR T don't do as well, but we usually save it for afterwards. We do use talquetamab if we're desperate in somebody who really has terrible disease to knock down disease burden. There was a paper published showing that cilta-cel after original ide-cel does work. So patients who were originally on a study. So it's not as efficacious, but there is at least a possibility of doing a second CAR, although the issue was going to be funding for that. We do collect stem cells. I didn't talk too much about that, to treat cytopenias after treatment. And there are a variety of other factors too that contribute to toxicity and inferior outcomes. We need to improve bridging. For example, where there's some studies from Europe showing that irradiation of essentially spot welding of high disease burden seems to benefit patients right before CAR T cell therapy. This is while the cells are being manufactured. We know that poor patient performance status is a bad factor for outcome. We need to decrease tumor burden during bridging because of the risk of toxicity and early progression, and decrease inflammatory states. We do know that patients who have an autoimmune disorder and who are receiving therapy for that, trying to do a CAR T cell in those patients is quite difficult because of the concern that you'll turn an autoimmune cell into a CAR T cell. And we also need to control disease, which predisposes to BiCans. And so can other therapies such as bispecifics be alternatives, as I mentioned earlier? So I think this will work. Let's see. So, sorry. I like to share that one because, whoops, I have to now figure out how to turn it off. Sorry. So I like to point that out just because it's funny. And number two, we do have the whole possibility of using a variety of things that potentially would improve how we do CAR T cell therapy. And I do have time, so I'm going to show you this as well. This is from a Blue Sky, which I'm on more than Twitter. It's a little less toxic. And so this is let me turn slideshow. Sorry, I wasn't sure if I'd be able to use this. And of course, now it's not letting me go into slideshow mode. There we go. Here's the T cell. I like the music. And there's a cancer cell that's attacking in punching holes with perforin and injecting granzyme B. And the T cell keeps hitting the cancer cell with multiple hits. And the cancer cell can't take it anymore and shrivels up and dies, which should occur any minute. Yes, and the T cell wanders away. So this is something that I could have only dreamed about when I was a fellow. It's pretty amazing that we now have this technology. So let me stop this. And I will get out of this. I'm going to stop sharing and I'll share again in a sec with my thank yous, which are, here we go, from current slide. And I have to share again. Oh, I have to get. All right, I'm sorry, I've lost my Teams. So I will get my Teams back and so you can see me. So I apologize for, there we go.
Thank you,
Almost
Philip.
there. Yeah, I just want to do my thanks because I have to I have to acknowledge all my colleagues. So I apologize for messing that up. And maybe I'll just stop there and I'll figure out how to share. There we go. So I want to thank everyone who was involved with my group. As you can see, I'm technologically a little challenged here. There we go. And my big thanks are to my colleague Heman Hamadpour, who Vitaly mentioned earlier. Heman has really made a huge difference in my ability to do basic science research. And this is a picture, if I can ever get it to work, of, there we go. As you can see, I am very technologically challenged when I have two presentations I'm trying to do. I should have stuck them all together.
Okay,
Finally.
oh, yeah.
There we go. So Heman's here. And that's Jens Hillengass, who's a big myeloma guy, and we're very lucky to have at Roswell. And these are all the people in his lab who are involved with working. Currently now, he's trying to make a better CAR T cells. So we're hoping to have something available within the next couple of years. And I will stop there and say, this is what therapy for relapsed malignancies looked like in 1975, and this is what we hope they'll look like in 2030. And now I will officially stop sharing. Thank you for listening.
Thank you, Phillips, again, for a wonderful presentation, a very good summary. So, audience, if you have any questions, you can raise your hand and you can type your query, and I'll try to read it for Philip. The other thing is, in the meantime, I'll ask a question myself. You clearly showed that cellular therapies are moving towards more front lines, and the companies are making more accessible and more easy to go. They're removing the observation times, the hospitalization times. But we still, like in our, in this country, in Ireland, we do still have very limited access to CAR T cell therapies. We are only still a cell approved for myeloma in October last year, but access of the patient across Ireland is very poor in general. So, and again, the limitation is because it's autologous CAR T. What do you think? So, what's the way to go? I'm thinking about myeloma, only thinking also about other applications for cellular therapies.
Yeah, I mean, that's why the allogeneic one initially had great appeal, because you just pull it out of the freezer. The issue is, how much is it going to cost? Although it can be scaled to a large number for manufacturing, which is appealing. And theoretically, you can do multiple dosings. Poseida has been doing multiple dosings of their allogeneic CAR. Then along comes the in vivo CARs, which are also something you can just pull out of the freezer and thaw up and just inject into a patient. There's no manufacturing. And the fact that J&J has partnered now with Colonia, which is a company based in Massachusetts, is very interesting, because I think they see the potential future there. These are also going to expand, and hopefully the price will come down, because if this is used, if this type of therapy is used for diseases such as autoimmune disorders, that will expand indications. As you know, our colleagues from Europe have done a fair amount of study in patients with horrible autoimmune disorders using both CD19 and BCMA-directed CAR T cells with success. Whether or not this scales is not clear, but if it scales using an in vivo CAR, that would make it, I hope, much more accessible for patients. As you know, pharmaceutical companies are beholden to stock shareholders, and so that is a major issue, that capitalism does drive this, but I would like to see the costs come down, because it makes it much more accessible for patients to have this. And I'm thinking, if it works, in vivo CARs or allogeneic CARs may be the way of the future as opposed to autologous, but that's just speculation on my part.
And just again, in terms of delivery, or in delivery of the specific antibodies, every time the new immunotherapies come to market, we're all concerned about safety. But with the time we get over this probably more psychological barrier, and we learn also how to treat the patients a bit better, even in outpatient setting. So do you have any outpatient treatment program with specific antibodies or CAR Ts? Because I know few people in the US, they do have it.
We have both. The bispecifics in particular, they were originally done on the cell therapy service. And then we realized this is easy. And so it moved over to our myeloma service and our lymphoma service. And those patients are seldom hospitalized. They may be, I should say, they may be for the initial therapy, a day or two, and then they're discharged. Or they're done as an outpatient because it's pretty well tolerated, and they're never hospitalized. So that's been a big shift. For CAR T, it's a little different. It's a sort of a hybrid where the patient gets the LD chemo unless there's major issues. Then they're admitted to the hospital for the infusion, and then kept for 1, 2, or 3 days, depending on toxicity. And if they develop, and if their discharge develop toxicity, then they're readmitted. So we've actually cut down on the days of hospitalization quite dramatically. I think the average is about 5 or 6 days, and that includes patients who then get admitted and are here for an extended treatment for, say, ICANS. So it's a lot smoother than it was because we've gotten a better understanding. And for the lymphoma ones, our team has a little bit easier time of it. Cilta-cel still a little bit tough. And as I mentioned earlier, there is the problem of late toxicity with the motor neuron disease. We had a run of about 3 Bell palsies and sort of this Parkinsonian-like affect in some of the patients. Some of them became somewhat flat. So far, our patients have all recovered, unless they have progression of disease, from the motor neuron disease. I currently have one now we're trying to work up. His MRI so far is normal, and he's going to get a lumbar puncture to look for the CARs, and then the decision we made, do we give him steroids or not? Because he doesn't think there's a problem, but his wife notes that he's quite flat. So, and this is several weeks after the CAR T. So that's something that I know Janssen is sort of dealing with, but at least the majority of patients seem to resolve that complication.
Okay. I don't see any questions, so just ask the last question. So it's about tumor microenvironment. So and hematological toxicity of CAR Ts. I believe with cilta-cel from the original trial from the CARTITUDE-1, there was reported something like nearly 10% of patients getting MDS. So there is probably clear interaction between the myeloid cells and multiple myeloma tumor cells. So what's your view on that? I know you're working a lot on tumor microenvironment. So what was the relationship between both you're seeing now?
Oh, that's a big question. I just had, when I used to do everything, one of our patients, she had her axi-cell for resistant lymphoma back in, when was that, 2018. And she just developed MDS. And so, not clear, because she also got our hyper-CVAD as part of her therapy. And then, so it's not clear. She had prolonged cytopenias. So she was getting GCSF twice a week for until she developed MDS because she had neutropenia. And initially she got marrowed like 5 different times over several years, nothing. And then all of a sudden MDS. So no, you bring up a good point, is how much of it is damage from the prior therapy, which then eventually rears its ugly head, versus how much of it is MDS in an inflammatory microenvironment, because it does appear in patients who have persistence of their CAR T that there is inflammation, which predisposes to things like cancer. How this all may occur is not clear. We also have had, interestingly, there have been tumors that are CAR T cell tumors. They're T cell cancers, although the one that we most recently reported, this fellow developed a lesion on his scalp. It was resected completely by the dermatologist, and it was a T cell lymphoma that had the CAR. He had a complete workup, nothing, and he received no further therapy. So it was weird. And then there have been other cases where it's full-blown systemic lymphoma. So ranging from MDS to lymphoma, there's a lot we don't understand. We're potentially interested, as mentioned in your introduction, thank you, that we're looking at CXCR2 inhibition because it inhibits inflamed neutrophils, which are also very immunosuppressive. It was funny, I was just looking, one of my colleagues at Sloan Kettering is also using CXCR2 inhibition to actually modulate craniospinal radiation. So because of its anti-inflammatory effects. So there's a lot of stay tuned. I know that was a very long-winded answer for something I'm not completely sure about in terms of how this develops and why is it developed in some patients and the majority of patients don't get these diseases.
Okay, thank you very much, Philip. So thank you very much for your presentation and for the audience to join today's UCC Cancer Seminars. And just before we end, so to announce next UCC Cancer Seminars, which is going to be on the 11th of May. On the topic of surgery, will be presented by CH Surgical Lead, Noel Lynch. Thank you, everybody, again, and have a good rest of the day. Thank you, Philip.
Thank you very much. Appreciate it.
Bye-bye.